Hi my friends, in this video I will talk about cell organelles or cell structure. Because we have many organelles, I will divide them into two videos. This is the first one. Let's start. Let's start in this video. In front of you the cell of human or animal. It's called an animal cell, and it's an eukaryotic cell, by the way, because it contains its own nucleus. Okay, I will make it obvious or more clear for you by pulling the transparency of it so that you can see the nucleus alone here. Let's move to that nucleus and let's describe it together. First of all, you should understand that the cell works as a factory, and for every factory there should be a manager or man in charge control office. This control office, what do you think it is? It's a nucleus. Okay, look carefully to the nucleus in front of you. We'll find that it's surrounded by a double membrane, letting some holes or pores in it to allow the exchange of materials from inside the nucleus to outside it and vice versa. It's okay. Let's zoom together here. Look, this is a double membrane. You see here, this is a double membrane. It looks like that. Here, a double membrane, and it fused together, forming a hole. That one also a membrane, but it's a double membrane, so this hole is obtained by that way. Let's move to the original size. If we talk about the nucleus, it controls all processes, all activities, and also cell division. So it's responsible for controlling everything inside the cell. One of the most famous experiments that carried out on the nucleus, when the scientists removed the nucleus of an amoeba and left it without a nucleus, it died in few hours. So you can understand that the cell, the whole cell, will die without a nucleus in few hours. The first important point for nucleus, that it contains the genetic material in the form of thread-like structures in front of you, and they are called chromatin. Let's remove this figure first. You should understand that this chromatin is similar exactly to something called chromatid or chromosome. So chromatin, chromatid, chromosome, all of them are called the genetic material, but according to the lifetime or a specific sequence or chronological order or timing inside the cell, it takes three different shapes: chromatin, again chromatid, and chromosome. Let's see together what is the difference between them. During the time of rest, the cell eats and grow, feeds and grow and take and get rid of some wastes. So it's a vegetative or vegetation activity. It grows a little bit. So this genetic material takes the shape of chromatin in front of you. But after that, when the cell undergoes or prepare itself for cell division, this chromatin shorten and thicken to take the shape of chromatid. How can you draw the chromatid? Chromatid is like that. In fact, it has a double DNA inside it. Okay, and we usually draw chromatid as a single rod shape like that. In fact, there is no boundary for the chromosome, just for illustration. So this is called chromatid. During a phase called S phase, okay, this chromatid makes another exact copy of it. So we can make it like that, and they are connected in one point called centromere. The chromatid, and this is another chromatid, and they are together called chromosome. So every one of these chromatids will go to one of the two dividing or produce daughter cells. It's okay. So the first important for the nucleus that it contains the genetic material in the form of chromatin. What do you think this dark body inside the nucleus? This dark body inside the nucleus is called nucleus. Nucleolus is usually one, but sometimes or some cells have more than one nucleolus. So they are called nucleoli. So the singular is nucleolus and the many are called nucleoli. You will remove US and put just I. So let's finish the function of the nucleolus. Nucleolus is a dense, tangled piece where the chromatin or genetic material collected strongly around itself. In this nucleolus, two important organelles are being synthesized. What are they? So the nucleolus help in building ribosomes and RNA. Ribosomes and RNA. Ribosomes are dark spheres like that. They go outside the nucleus to the cytoplasm. Why? Because the ribosomes will help in building proteins. Here we should mention something like a mutualistic relationship. One builds another thing and the other thing will build the same first one. How? Inside the nucleus, this nucleolus here, nucleolus, will help in building ribosomes. Why? Because this area is rich in 1,000 copies of proteins. So the proteins here help in building ribosomes. But outside of the nucleus, this ribosomes will help in building proteins once more. Nucleus. So we said that the biggest organelle, the most obvious one, is the nucleus. We finished the nucleus, it's okay. Let's move now to another organelle. Here in front of you, this is a plant cell. By its characterizing shape, it has a hexagonal shape like that due to the thick cell wall. Okay, but look here at the center, it has a very big vacuole. It takes the whole space or the whole volume of the cell, or more than 50%. Okay, this organelle is called, what do you think the function of the vacuole for the plant cell? And why do the animal cells do not have these big vacuole like that? You should understand that the plant does not move from a place to another, okay? So it has to store something for the future, something for tomorrow. So it will store everything for the cell, like what? This vacuole will store water. Here, because we talk about the vacuole, we should mention that the animal cell has small vacuoles and they are called vesicles. Usually our vacuoles are used for transmitting materials within the cell, okay, or for secreting some substances outside of the cell, or for transmitting food molecules or food particles inside the cell, and they are called vesicles. Here we should mention amoeba. Amoeba is a unicellular organism that live in fresh water. So this is its nucleus. It lives inside the fresh water. The water will move into it by osmosis. So it's a big problem. Why? Because by passing time, this amoeba may explode. So it has to get or to push this water outside of it by collecting these tiny molecules of water and into a bigger vacuole and to a bigger vacuole until it go to the surface of the cell so it can explode. What do we call it? It's called contractile vacuole. Here in cleaning up organelles, we will talk about lysosomes. Look, where are the lysosomes? The lysosomes appeared now. Here, 1, 2, 3. You should know that the word some means a body. Lysosome, for example, means analysis or analytic. Why? Because they contain digestive enzymes or hydrolytic enzymes. These lysosomes help get rid of strange bodies or microbes. It has some digestive enzyme for digesting some food molecules that may leak into the cell or absorbed by the cell. The question now, why these lysosomes do not digest themselves from inside? Why this digestive juice will not affect their double membrane? Because it's maintained by a thick layer of carbohydrates. It's okay? Due to the heavy layer of carbohydrates. We should mention also that bacteria engulfed by white blood cells are digested by the same way. So you should know that the white blood cells contain large number of these lysosomes. For example, if we draw a white blood cell like that, it has a nucleus and it has large number of these lysosomes that contain digestive enzymes. That's okay. Why? Because this white blood cell will engulf or will swallow some microbes like that, and like bacteria, these bacteria can be digested by these lysosomes. Another point you should mention beside lysosomes, what are they? Peroxisomes. They do not contain digestive enzymes like lysosomes, no, but they contain oxidizing enzymes, help in changing harmful substances into less harmful ones or less toxic ones. Our time with the organelles that support the cell. Although the cell is very small and extremely small, but it still needs a support. Okay. This support can be maintained by two different types of organelles. The first one is called microtubules and the second one is called microfilaments. Let's compare between them and let's see them firstly. Look here, this is a microtubule. It takes the shape of tubes like that. You see this tube? Okay, you see this tube? That one? Okay, and they are called microfilaments. Like that. Let's make a whole comparison between them in the next layer. Let's start. Microfilaments looks like that. They are fiber-like structures, unlike the microtubules that has thicker body like that. Many of it. They are called microfilaments and that are called microtubules. The type of protein, they are made up of a protein called actin protein, while here tubulin protein. The microfilaments help in motion, help in movement. So they are involved in the muscle structure and they help in movement of amoeba. This amoeba cell, it has a nucleus and it lives in fresh water and it has to push some thread-like structure made up of actin, so these are called microfilaments. And in fact, it's called pseudopod, means false foot. By the way, microfilaments and microtubules together are called cytoskeleton. Okay, but let's return to the microtubules. Microtubules help in building cilia, flagella, centrosome, and spindle fibers. Let's see one by one. Look here. This is a tiny living organism. It's called Paramecium, and it moves by the help of this cilia in front of you. And this is another unicellular organism. It's called Euglonia, and it has a flagellum. And the many are called flagella. They are called cilia, and the singular is called cilium. Cilium, cilia, flagellum, flagella. That's okay. They are built above these tiny microtubules, that and that. So what about the centrosome and the spindle fibers? Let's go to another layer. Here, if you consider that this is a cell, like that, this centrosome 1, 2, 3, 4, 5, 6, 7, 8, 9, and here 1, 2, 3, 4, 5, 6, 7, 8, 9. By the way, same thing happens here. 1, 2, 3, 4, 5, 6, 7, 8, 9 in a circle. After that, during the cell division, this one will produce spindle fibers. These also are called spindle fibers. These spindle fibers, by the way, consist of tiny tubules, and so up and down like that. Look, this is a chromatid. It's being pulled down, and this is the other sister chromatid. So they have a very important function during separation of the chromatid or during the formation of spindle fibers up and down. Let's compare between the microtubules found in cilia and flagella and that one in spindle fiber, centrosome and spindle fibers or spindles. Here, if you have a cross section in a cilium or in a flagellum, you will find a circle consists of 2, 3, 4, 5, 6, 7, 8, 9, plus a pair in the middle. So this structure is called 9 plus 2. 9 means 9 pairs of microtubules plus a pair in the center of the circle. Their structure consists of 1, 2, 3. Count 1, 2, 3, 4. 1, 2, 3, 4, 5. 9 triplets. So 9 triplets. Let's compare. Here 9 times 3 equals 27 microtubule, while here 9 pairs, but it's written in this formula, 9 plus 2. You know that 9 means 9 pairs plus 2. I mean 9 pairs plus a pair in the center. This is the structure of cilia and flagella, while this is the structure of centrosome and spindles. 9 plus 2 or 9 pairs plus 2, so 18 plus 2 equals 20, while here 9 times 3 equals 27. By reaching that point, finish the first part of the cell structure. I wish you the best of the best and I wish you good luck. Thank you and see you soon. Goodbye.